We know the reactions catalyzed by an astounding variety of enzymes, turning tens of thousands of different substrates into a similar number of products. It turns out, though, that the types of reactions being catalyzed can be categorized into a conveniently small number of classes. First, oxidoreductases. They catalyze, as the name says, oxidation and reduction reactions. Many of them retain older, non-systematic names, including dehydrogenase, oxidase, and so on. Two examples are lactate dehydrogenase, which reversibly oxidizes lactate to pyruvate using the coenzyme NAD+, and ascorbate oxidase, which uses molecular oxygen to metabolize vitamin C or ascorbate. Transferases catalyze group transfer reactions, usually via a covalent intermediate of the substrate with the enzyme or a coenzyme. Here, for example, alanine transaminase transfers an amino group from alanine to alpha-ketoglutarate. Another example is one of the hard-working enzymes that detoxify foreign compounds or xenobiotics. Glutathione S transferase attaches a large variety of compounds to the thiol group in glutathione. Hydrolases, unsurprisingly, catalyze hydrolysis reactions. They are, in a way, specialized transferases that use water as one of the acceptor molecules, and they cleave a whole range of different bonds. Hydrolases are the largest of the 6 enzyme classes. About 1 in 3 known enzymes is a hydrolase. Typically, they are named after the substrate that is being hydrolyzed. So proteases, lipases, and esterases, which in turn include phosphatases and nucleases like DNA and RNA cutting enzymes, are all hydrolases. Here, for example, a peptide bond is cleaved by hydrolysis, and you can see which part of water has ended up in which product. Lyases are a little more difficult to understand. These enzymes catalyze the cleavage of a substrate, leaving behind a double bond. It is important to note that this elimination reaction involves neither oxidation nor reduction, nor hydrolysis with water. Some of the more famous lyases include enzymes labeled decarboxylase, aldolase, and dehydratase, which eliminate CO2, an aldehyde, or water respectively. For example, pyruvate decarboxylase catalyzes the elimination of carbon dioxide from pyruvate. This is no redox reaction, but a carbon-carbon bond is split, and a double bond is formed to produce the CO2 molecule. Some lyases catalyze the reverse reaction, the addition of one substrate to a double bond in another substrate. These are sometimes called synthetases. Isomerases catalyze structural change within a single molecule. A large number of different kinds of isomerizations are possible enzymatically, be it the conversion of one stereoisomer into another, the moving of a double bond or a group within a molecule, or a change in ring size, all without oxidation or reduction and without changing the molecular formula of the substrate. A simple example of an isomerase is alanine racemase. The final and smallest family of enzymes are the ligases. These enzymes join two substrates, forming a covalent bond. Here, for example, glutamine synthetase ligates ammonia to glutamate to form glutamine. As another example, DNA ligases are an essential tool in the molecular biology lab to covalently join up two strands of DNA. Crucially, unlike lyases or transferases, ligases need energy from ATP to perform this reaction.